Good morning, everyone. Today I'm going to talk about the physiology of neuromuscular transmission. First, let's start with the objectives. The first objective is to describe the physiological anatomy of the neuromuscular junction. Then describe the sequence of events occurring during neuromuscular transmission. Then I will explain the process of exocytosis and the concept of Quanta release of the neurotransmitter and its fate. And here we are talking about acetylcholine. And then I will explain the generation of the end plate potential and the safety factor. Objective 5, to explain how the neuromuscular transmission may be blocked. And I will list the drugs which affect this transmission. And finally, I will briefly describe the pathophysiology of myasthenia gravis and the Lambert-Eaton myasthenic disease or Lambert-Eaton myasthenic syndrome. Action potential is generated and conducted in neurons and muscle cells, and we call neurons and muscle cells excitable cells because they are able to generate and conduct action potential. The signal is communicated from cell to cell, either from neuron to neuron or from neuron to a neuroeffector cell, which could be a muscle or a gland, through intercellular junctions called synapses. Therefore, a synapse is a junction between cells that is specialized for cell-to-cell signaling. Approximately, we have about 100 billion neurons in the human brain, and each neuron will give about 1,000 branches. And each branch will end in a small swelling called the presynaptic terminal or synaptic button. If we put all these numbers together, we can see that the human nervous system has an order of 100 trillion synapses, or 10 to the power 14 synapses, and this is just the nervous system, and if we add other synapses elsewhere in the body, the number will be much higher. Synaptic transmission can be either electrical or chemical. Here we can see the two types of synapses. On the left side of the slide we can see the chemical synapse. On the right side we see the electrical synapse. A chemical synapse is a synapse where the transmission of signal is mediated by chemicals released from vesicles. Inside these vesicles, the chemicals are stored, and once an action potential arrives to the presynaptic terminal, the neurotransmitter will be released. It will cross the cleft or the synaptic cleft or space between the presynaptic terminal and the postsynaptic terminal or the neuroeffector cell and bind to the receptor on the postsynaptic membrane to produce an action. On the other hand, in the electrical synapse, it is a synapse where there is electric current flow between the presynaptic neuron to the postsynaptic neuron through gap junctions. So the difference here between chemical synapses and electrical synapses, in chemical synapses we have a delay. So if we measure the membrane potential in the presynaptic neuron here, the black line, we can see that the action potential arriving to the presynaptic neuron, this will lead to an excitatory postsynaptic potential in the postsynaptic neuron after a period of delay. This is the time needed for calcium to enter, for the exocytosis of the neurotransmitter from the vesicles, the time needed for the neurotransmitter to cross the synaptic clefts and reach the receptors on the postsynaptic membrane, and then the opening of the postsynaptic membrane ion channels, and then to produce this excitatory postsynaptic potential. On the other hand, because current flows very rapidly through the gap junction, there is a very short delay in the case, or no delay in the case of electrical synapse. As you can see here, this is the action potential in the presynaptic terminal, and this will lead to an action potential in the postsynaptic neuron with hardly very short delay. So at electrical synapses, current flows directly from one cell to the other cell through gap junction channels. And these gap junctions, as you can see here, the synaptic cleft is very narrow, about 2 to 4 nanometers, on average 3.5 nanometer. This area is full of gap junctions. Each gap junction contains or is composed of 2 hemichannels called connexons, and each connexon is composed of 6 subunits called connexins that surround a water-soluble pore that will allow the flow of ions between the 2 cells. Electrical synapses are designed for rapid and synchronous transmission. We saw in the previous slide there is very short delay or almost no delay. And most electrical synapses are bidirectional. So signal can be transmitted from either one cell, from either one of the connecting cells to the other. So the signal can pass from this side to this side or in the opposite direction. Most synapses in our body are chemical synapses, in which the presynaptic terminal contains many synaptic vesicles that are filled with high concentrations of the neurotransmitter. An action potential in the presynaptic nerve terminal releases neurotransmitter molecules that diffuse across the synaptic cleft and bind to receptors in the postsynaptic membrane. Receptor activation can either generate electrical or biochemical signals in the postsynaptic membrane or in the postsynaptic cells, and this electrical could be excitatory or inhibitory postsynaptic potentials. The neuromuscular junction, which is the topic of today's lecture, is an unusually large chemical synapse. So the neuromuscular junction is a chemical synapse that is responsible for the initiation of action potentials across the muscle's surface or the muscle membrane, and ultimately this will lead to muscle contraction, the excitation contraction couple. Now let's talk about neuromuscular junction. All skeletal muscles are under voluntary or reflex control by motor neurons. A motor nerve fiber divides into several branches as it approaches a muscle. As we can see here, this is a motor neuron that divides into several branches, and each branch will end into an axon terminal that will contact a muscle fiber. Each branch of the motor nerve fiber innervates one muscle fiber or one skeletal muscle fiber, usually at its middle or the middle of the fiber. A single motor neuron and all muscle fibers it innervates are called a motor unit. So this is a single motor neuron, and these are all the muscle fibers that are innervated by this single motor neuron. So we call this structure a motor unit. The junction between a motor nerve fiber and a muscle fiber is known as a neuromuscular junction, and as I said, this is a chemical synapse. Here we can see the physiological anatomy of the neuromuscular junction. The neuromuscular junction is composed of presynaptic structure and the postsynaptic structure separated by a synaptic cleft. The presynaptic structure is a branch of the somatic motor neuron that expands at its end to form the axon terminal or the synaptic knob. The synaptic terminal contains a lot of synaptic vesicles that are packing acetylcholine inside them. As you can see, the vesicles or the synaptic vesicles are clustered at certain areas or aggregate at certain areas near the presynaptic membrane or specific areas of the presynaptic membrane. We call them the active zone. So the synaptic vesicles are not diffusely scattered near the presynaptic membrane, but they are gathered at certain specific points. We call them the active zones. As you can see that the synaptic cleft is sealed by Schwann cells. The synaptic cleft contains a peptidoglycan or extracellular matrix that covers the muscle and also extends into the synaptic cleft. It is a basal lamina or an extracellular matrix that is rich in the enzyme acetylcholinesterase that breaks down acetylcholine after it performs its function. As you can notice also that the synaptic terminal rests on a depression in the postsynaptic structure or in the muscle. This is called the synaptic gutter or synaptic trough. And the part of the skeletal muscle membrane that contributes to the synapse is called the motor end plate. It is thrown into folds to increase its surface area, and it is very rich in the acetylcholine receptors, especially at the top of the folds. These folds are called the subneural or junctional folds. Here, in a more clear figure, we can see that, again, this is the synaptic terminal. It is rich in mitochondria, also microtubules for the transport or the axonal transports of the vesicles. Vesicles are gathered or clustered near specific areas of the presynaptic membrane we call the active zone, which is the place where the synapses, the vesicles fuse with the presynaptic membrane to release acetylcholine. Again, we see the basal lamina that is rich in acetylcholinesterase and the motor end plate that is thrown into folds called the subneural folds. The folds will create synaptic or primary and secondary postsynaptic clefts, and this will further increase the surface area. You can see here that we have large number of acetylcholine receptors in red, and the basal lamina, as can be seen here, that is rich in acetylcholinesterase. It is also here, the figure is simplified. This basal lamina is also thrown inside the primary and secondary clefts, so it will or the acetylcholinesterase that is present in that basal lamina will destroy acetylcholine after it performs its function. So each motor neuron runs without interruption from the ventral horn of the spinal cord or medulla to the neuromuscular junction as a large myelinated axon. As the axon of the motor neuron approaches the muscle fiber, it loses its myelin sheath and divides extensively into several fine branches called axon terminals, as we can see here. Each terminal is expanded at its end to form a knobby structure called terminal button, synaptic knob, or end foot that makes a single point of synaptic contact with the skeletal muscle fiber, as I said, midway along the length of the skeletal muscle fiber. The synaptic vesicles are clustered around specific points near the presynaptic membrane called the active zones. As I said here, the synaptic vesicles are not diffusely or randomly distributed near the presynaptic membrane. They are present near specific or clustered near specific areas of the presynaptic membrane called the active zone, as we can see here in this figure. Now let's talk about the presynaptic portion of the neuromuscular junction. Then we'll talk about the synaptic cleft, and then later on about the postsynaptic portion of the neuromuscular junction. The synaptic vesicles are synthesized in the rough endoplasmic reticulum of the nerve soma. So they are produced here in the rough endoplasmic reticulum in the nerve cell body. Then they are processed or directed to the Golgi complex, where they undergo processing, maturation, and sorting, and then they will be transported to the axon terminal by the microtubule system. So the synaptic vesicles are synthesized in the rough endoplasmic reticulum of the nerve cell body and are then directed to the Golgi complex or network, where processing, maturation, and sorting occur. They are transported to the nerve terminal, which contains around 300,000 vesicles. So each nerve terminal at the neuromuscular junction contains about 300,000 vesicles by a fast axonal transport mediated by the microtubule system. Also, the vesicles can be recycled in the opposite direction back to the rough endoplasmic reticulum, as we'll see later on in the recycling of the synaptic vesicles. So the axonal transport is bidirectional. I want to attract your attention to one difference. If we are talking about peptide neurotransmitters, usually they are produced in the cell body inside the vesicles, and the vesicles that contain the neurotransmitter are then transported axonally to the nerve terminal. In the case of non-peptide neurotransmitters like acetylcholine at the neuromuscular junction, the vesicles are produced in the cell body and then transported empty to the axonal terminal, and then they will be filled with the acetylcholine that is formed in the cytoplasm of the axon terminal. So in the case of neuromuscular junction, the vesicles arrive to the terminal empty, and then they are filled with acetylcholine in the axon terminal. As I said, the vesicles are filled with the neurotransmitter acetylcholine at the axon terminal. And let's have a closer view. Here we have the membrane of a synaptic vesicle. The membrane is rich in the proton pump. That is an example of primary active transport system. This proton pump will generate high concentration of hydrogen ions inside the synaptic vesicle. Now we have the cytoplasm here outside the vesicle, where choline will react with acetyl-CoA in the presence of choline acetyltransferase, the enzyme responsible for the production of acetylcholine from choline and acetylcholine. And now we have ready acetylcholine that will be transported or antiported in exchange for hydrogen. So acetylcholine will enter the synaptic vesicle at this exchanger in exchange for hydrogen down its concentration gradient. As I said, hydrogen concentration is very high, so hydrogen will operate the secondary active transport system. Therefore, acetylcholine is synthesized locally in the cytoplasm of the nerve terminal from acetyl-CoA and choline by the enzyme choline acetyltransferase. Then acetylcholine is rapidly transported into the synaptic vesicle via the acetylcholine hydrogen exchanger, and it is stored there until it is released by exocytosis. Each vesicle is filled with around 10,000 acetylcholine molecules. So in most textbooks, the range of or the number of acetylcholine molecules inside one synaptic vesicle is between 5 to 10,000 molecules. Now let's talk about the synaptic cleft. The synaptic cleft is the gap between the terminal button and the muscle fiber or the skeletal muscle fiber, which is about 20 to 50 nanometer wide, and in some neuromuscular junctions it can be up to 100 nanometer wide. This cleft is filled with the extracellular fluid and the basal lamina that is composed of extracellular matrix, and this synaptic cleft is sealed all around by the Schwann cells. The muscle fiber is covered by basal lamina consisting of collagen, glycoproteins, and other extracellular matrix proteins, and this is the basal lamina. This basal lamina also extends into the synaptic cleft and into the primary and secondary clefts or folds, and as I said, it is very rich in the enzyme acetylcholinesterase. So the synaptic basal lamina contains high concentration of the enzyme acetylcholinesterase that terminates the transmission by rapidly hydrolyzing acetylcholine to choline and acetate. Now, the postsynaptic portion of the neuromuscular junction. The part of the sarcolemma or the skeletal muscle cell membrane that lies directly under the terminal button or the synaptic knob is known as the motor end plate or end plate membrane. The terminal button lies in the groove or invagination of the sarcolemma called the synaptic trough or synaptic gutter, as we have seen in one of the previous slides, on the surface of the skeletal muscle fiber. At the bottom of the gutter are numerous smaller folds of muscle membrane called junctional or subneural folds, and they are primary and secondary clefts that contain numerous acetylcholine receptors. The subneural folds increase the postsynaptic membrane surface area and allow the accommodation of large numbers of acetylcholine receptors. The active zone of axonal terminal is located over the subneural folds, as we have seen in one of the previous slides. So what are the steps that happen during the neuromuscular transmission? First of all, an action potential should arrive through the motor neuron or the somatic motor neuron, and this electrical activity or this change in the membrane potential will open voltage or voltage-gated calcium channels. The voltage-gated calcium channels are usually concentrated around the active zone. So the calcium entry there will increase the calcium concentration in the microdomain just around the active zone. The increased intracellular calcium near the active zone will cause the fusion of the synaptic vesicles and the exocytosis of acetylcholine into the synaptic cleft. The released acetylcholine will bind to the ligand-gated receptors on the postsynaptic membrane or the motor end plate, 2 acetylcholine molecules per receptor. This will open the ligand-gated ion channel, which is permeable mainly to sodium. Sodium entry into the skeletal muscle fiber at the motor end plate will generate an end plate potential that will bring the sarcolemma to threshold, and this will open voltage-gated sodium channels, and this will lead to the propagation of action potential across the sarcolemma down into the T-tubules to start the excitation contraction coupling. Then acetylcholine will be degraded by acetylcholinesterase that is present in the basal lamina, which is there in the synaptic cleft. The exocytosis of acetylcholine vesicles from the motor nerve terminal is mediated by the interaction of different membrane proteins called the SNAR membrane proteins. This is a family of membrane proteins. Those membrane proteins are present, some of them are present in the synaptic vesicle membrane, and others are present in the presynaptic membrane. For instance, one of the SNAR proteins is the synaptoprevin, which is present in the synaptic vesicle membrane, and we have another 2 SNAR proteins. These are the syntaxin and SNAP25. The interaction between them will prime the synaptic vesicle and prepare them for fusion with the presynaptic membrane to open through a pore to release the acetylcholine that will diffuse across the synaptic cleft to bind to acetylcholine receptors. So acetylcholine release from the synaptic vesicles is mediated by SNAR proteins. During depolarization and calcium entry through the voltage-gated calcium channels, SNAR proteins on the vesicle, which is synaptoprevin, and SNAR proteins on the presynaptic membranes, mainly SNAP25 and syntaxin, they interact and the vesicle is attached to the presynaptic membrane in preparation for the fusion of the synaptic vesicle with the presynaptic membrane. and upon depolarization and calcium entry, calcium binding to the calcium sensor, which is synaptotagmine, one of the synaptic vesicle membrane proteins and also a member of the SNAR proteins, triggers fusion of the vesicle to the nerve membrane at the active zone to release its contents of acetylcholine into the synaptic vesicle. Here we can see in this figure that we have a synaptic vesicle membrane, the plasma membrane of the axon terminal or the presynaptic membrane. We have the synaptoprevine, we have the syntaxin, and we have SNAP25, as well as other SNAR proteins, and the interaction between them will prepare for the fusion of the synaptic vesicle with the presynaptic membrane, especially after the entry of calcium and its reaction with the calcium sensing SNAR protein, which is synaptotagmine. So here we can see a full cycle how things happen. First of all, the synaptic vesicles that are filled with acetylcholine become docked. This is called docking, where the synaptic vesicles are clustered near the active zone due to the interaction of the SNAR proteins. And this will be followed by priming of the synaptic vesicle in preparation for its fusion with the presynaptic membrane once calcium enters through the voltage-gated calcium channel. Then the vesicle membrane will detach and will be recycled by endocytosis, and then the synaptic vesicle will be refilled with acetylcholine that is formed in the cytoplasm, and the process will keep repeating over and over. The synaptic vesicles, after releasing their content of acetylcholine during the process of exocytosis, are recycled. There are three different ways of recycling the empty synaptic vesicles. One of them is called the kiss and stay mechanism, where the synaptic vesicles empty their content of acetylcholine into the synaptic cleft. They remain docked in the active zone, just they will be refilled by the neurotransmitter and primed again in preparation for fusion in the presence of calcium with the presynaptic membrane. So they don't leave the active zone. They remain docked, and while docked in the active zone, they are refilled with the acetylcholine or with the neurotransmitter. Another mechanism of recycling the synaptic vesicles or the empty synaptic vesicles, the empty synaptic vesicles will be undocked in a process called kiss and run. So they will leave the active zone, then they will be refilled with the neurotransmitter acetylcholine away from the active zone, and then they will go through the docking again, then priming, then in the presence of calcium fusion to the release of acetylcholine, and then endocytosis, and the cycle will keep repeating over and over. The last mechanism is called the endosomal recycling, the release or after fusion of the vesicle with the presynaptic membrane and the release of acetylcholine. Endocytosis will be mediated by a protein called clathrin. So these blue bars indicate the protein clathrin that will help in the endocytosis of the empty synaptic vesicle. Then the clathrin protein will dissociate from the endocytized vesicle, and there are two fates. Either the empty synaptic vesicle will remain in the terminal, it will be refilled with this neurotransmitter acetylcholine, docked again, primed and prepared for fusion in the presence of calcium, or it will be transported through the axon back to the cell body, to the nerve cell body, to undergo endosome fusion and preparation again and processing in the Golgi apparatus, and then back through axonal transport to the nerve terminal, where they will be refilled with the acetylcholine neurotransmitter docked, primed, and will fuse again in the presence of calcium to release acetylcholine. So these are the three different ways of recycling of empty synaptic vesicles. Now, what is the fate of released acetylcholine? What happens to acetylcholine once it is released from the synaptic vesicles into the synaptic cleft? Acetylcholine is rapidly removed by two mechanisms. First one, the most common one, is destruction by the enzyme acetylcholinesterase that is located on the basal lamina in the synaptic cleft. Most acetylcholine is broken down by this mechanism. Another mechanism that has a minor role is diffusion. Small amounts of acetylcholine diffuse out of the synaptic cleft. So here we see the fate of acetylcholine once it is destroyed by the enzyme acetylcholinesterase. The choline will be actively transported back into the presynaptic or the synaptic terminal. Then, in the presence of the enzyme, acetyl-CoA will be combined with choline to produce new acetylcholine that will be transported through the acetylcholine hydrogen exchanger into the synaptic vesicle in preparation for the next cycle of exocytosis. So after acetylcholine acts on its receptor, it is hydrolyzed by the enzyme acetylcholinesterase into acetate and choline, as we can see here. Then choline is actively transported or reabsorbed into the nerve terminal to be used again for the formation of acetylcholine or new acetylcholine. The entire process of acetylcholine release, action on its receptor, and destruction or degradation usually takes about 5 to 10 milliseconds. So it is a very rapid process. Now let's talk about the concept of quanta release of neurotransmitters, including the quanta release of acetylcholine. In general, neurotransmitters, including acetylcholine, are released in discrete packets called quanta, which is the plural of quantum. What is a quantum? A quantum corresponds to the neurotransmitter content of a single synaptic vesicle. As I previously explained, I said at the neuromuscular junction, one presynaptic vesicle or one synaptic vesicle contains about 5,000 to 10,000 molecules of acetylcholine. So the quantum is about 5,000 to 10,000 molecules of acetylcholine, which is the content of a single synaptic vesicle. At the neuromuscular junction, about 5,000 to 10,000 molecules of acetylcholine are stored in a single synaptic vesicle, and again at the neuromuscular junction, approximately 100 to 300 synaptic vesicles or quanta are normally released in response to a single presynaptic action potential. What do we know about acetylcholine receptors? The released acetylcholine binds to acetylcholine receptors present on the postsynaptic membrane or the motor end plate, and these receptors are ligand-gated ion channels present on the postsynaptic membrane. As I said, acetylcholine receptors or one acetylcholine receptor is a heteropentamer. Pentamer means 5 subunits with a subunit composition of alpha 2, beta, gamma, and delta. So acetylcholine receptor contains 2 subunits or 2 alpha subunits, 1 beta, 1 gamma, and 1 delta. Each alpha subunit has 4 transmembrane spanning segments, as we can see here, and the entire receptor has 5 subunits. Acetylcholine binds to the alpha subunit, so we need 2 acetylcholine molecules per receptor because the receptor has 2 alpha subunits. The binding of acetylcholine to these receptors increases the sodium and potassium conductance of the postsynaptic membrane. As we can see here, the binding of 2 acetylcholine molecules to the binding site on the receptor will cause conformational change, and this will open the channel and will increase the conductance for sodium entry and to a lesser extent the potassium efflux. Sodium influx creates a local positive potential change inside the muscle fiber that is called the end plate potential. Acetylcholine released, or the released acetylcholine, is rapidly removed from the synaptic cleft through the enzyme acetylcholinesterase to prevent re-excitation of the receptor after the first action potential. As I said, end plate potential is a local graded potential and is analogous to an excitatory postsynaptic potential but much larger. All neuromuscular junction end plate potentials are excitatory and cause an action potential on the adjacent postsynaptic membrane every time an action potential arrives at the presynaptic membrane. One excitatory postsynaptic potential is normally more than sufficient to cause action potential generation on adjacent skeletal muscle membrane. Action potential is propagated on skeletal muscle fibers from the center towards both ends of the muscle fiber so that the center or the central sarcomeres contract first. As you can see here, we have this plot for membrane potential. We can see here this orange line. This is a graded potential in the postsynaptic membrane that is called end plate potential, and its amplitude usually is enough to bring the adjacent skeletal muscle membrane to threshold to initiate an action potential. Now let's talk about the concept of safety factor. Each nerve impulse releases about 100 acetylcholine vesicles, up to 300 acetylcholine vesicles, and each vesicle, as we said, contains between 5,000 to 10,000 molecules of acetylcholine. Actually, we need only 10 vesicles, or only 10 vesicles are required to bring the muscle membrane potential to threshold from -90 to -65 millivolt. Therefore, the released acetylcholine vesicles, about 100 vesicles, can activate 10 times the number of acetylcholine receptors on the postsynaptic membrane. So we have 10-fold safety factor. This means that the muscle action potential is always generated for each action potential that arrives at presynaptic membrane. So whenever an action potential arrives at the presynaptic membrane, the generated end plate potential will lead to an action potential in the surrounding skeletal muscle membrane. Now let's talk about some drugs that affect neuromuscular transmission. Some drugs will block the neuromuscular transmission while other drugs will enhance neuromuscular transmission. Among the common drugs or common toxins, we can see here the toxins that inhibit acetylcholine release, or tetanus toxin and botulinum toxin. Some drugs like tetrodotoxin and saxitoxin, they inhibit the neuronal voltage-gated sodium channels as well as the muscular voltage-gated sodium channels. Other drugs like omega conotoxin, they will inhibit the voltage-gated calcium channels. Here we have some drugs like physostigmine or neostigmine or DFP. They inhibit the enzyme acetylcholinesterase, so more acetylcholine will be available or present in the synaptic cleft, and this will enhance neuromuscular transmission. Another set of drugs that affect the acetylcholine receptors at the motor end plate, acetylcholine and nicotine are stimulatory or agonists for these receptors, while D-tubocurarine and alpha-bungaratoxin are inhibitory, or they block the acetylcholine receptors in the motor end plate. So there are several deadly toxins which block neurotransmitter release, and these include tetanus toxin and botulinum toxins, B, D, F, and G. They act on synaptoprevin, which is a SNAR protein associated with the synaptic vesicle membrane, while botulinum toxin C acts on syntaxin, which is a SNAR protein associated with the presynaptic membrane. On the other hand, botulinum toxins A and B act on the SNAR protein SNAP25 that is associated with the presynaptic membrane. All these toxins, they prevent the exocytosis or the release of acetylcholine from the synaptic vesicles into the synaptic cleft. There is one positive side of these toxins, where local injection of small doses of botulinum toxin, which is Botox, has proved effective in the treatment of a wide variety of conditions, such as the injection of Botox into the lower esophageal sphincter to relieve achalasia, to relax the sphincter and smoothen the swallowing process. Another useful use of Botox is its injection into facial muscles, so they will relax, and this will remove the wrinkles in the face. We have other drugs also that affect the neuromuscular junction transmission. We have a set of drugs that stimulate the muscle fibers by acetylcholine-like action, and these drugs include methacholine, carbachol, and nicotine. They have the same effect on the muscle fiber as does acetylcholine, but are not destroyed by cholinesterase, or are destroyed so slowly that their action often persists for many minutes to several hours. Another group of drugs that stimulate the neuromuscular junction by inactivating acetylcholinesterase that is present in the basal lamina. And these drugs are neostigmine and physostigmine. They inactivate acetylcholinesterase for up to several hours. The other drug, which is diisopropyl fluorophosphates, DFP, it inactivates acetylcholinesterase for longer periods, which is weeks. Third group of drugs that block transmission at the neuromuscular junction, and this is a group of drugs known as curariform drugs that can prevent passage of impulses from the nerve ending into the muscle, like D-tubocurarine, which blocks acetylcholine receptors on the muscle fiber or in the motor end plate. Now let's talk about two disease conditions that are where the neuromuscular transmission is affected. The first one is myasthenia gravis. It occurs in about 1 in every 20,000 persons. It is an autoimmune disease. Patients have developed immunity against their own acetylcholine receptors that are present at the motor end plate. The end plate potentials, due to the low number of receptors in the muscle fiber, are mostly too weak to stimulate the muscle fibers. So the end plate potentials will not bring the surrounding membrane potential to threshold to generate an action potential. If the disease is severe enough, the patient might die due to the paralysis of muscles, especially the paralysis of the respiratory muscles. We have different varieties of the disease. Some patients, they have only the extraocular muscles affected, so they have diplopia and ptosis, where the upper eyelid drops. And in another group of patients, they have generalized weakness of their muscles, and they have fatigue. The disease can be usually improved or ameliorated for several hours by administering neostigmine or some other anticholinesterase drugs. Another disease, which is Lambert-Eaton myasthenic syndrome. It is a muscle weakness caused by an autoimmune attack against the voltage-gated calcium channels in the presynaptic nerve endings. So the number of channels decrease, and this will lead to less calcium to enter into the nerve terminal and less neurotransmitter to be released. Usually in this disease, the proximal muscles of the lower extremities are primarily affected, producing waddling gait and also difficulty in raising the arms. About 40% of patients with Lambert-Eaton myasthenic syndrome also have cancer, especially the small cell cancer of the lungs. A syndrome similar to the Lambert-Eaton myasthenic syndrome can occur after the use of aminoglycoside antibiotics, because these antibiotics impair the voltage-gated calcium channels in the presynaptic membrane. With this slide, we come to the end of this session. Thank you for your patience.